Dear students, please note down. These notes are available in the description of video. Please download your notes. In today's lecture, we will clear mitochondria. Before this lecture, we have discussed nucleus, lysosomes. And in today's lecture, we will clear mitochondria, that is named as powerhouse of cell. This mitochondria, it was discovered by Colliker in 1850. But this term mitochondria, that was coined by Benda in 1897. This mitochondria, which is named as powerhouse of cell, this is a double membrane bound cell organelle. And in this mitochondrial matrix, that is the main center for the release of ATP energy. That's why it is named as powerhouse of cell. Here in this matrix, mitochondrial core matrix, or it is inner chamber, Krebs cycle takes place. Oxidative phosphorylation takes place. Note down, in chapter photosynthesis, we have discussed photophosphorylation. And in this cellular respiration, in mitochondria, oxidative phosphorylation means photophosphorylation occurs in chloroplast. Note down, in tomorrow's lecture, we will discuss chloroplast. But today we will discuss mitochondria. Here in mitochondria, oxidative phosphorylation means in presence of air, in presence of oxygen, breakdown of food, and that oxidation produce energy in the form of ATP or reducing agent, and the energy gets stored in the form of ATP. It means the mitochondrial matrix, it is main center of the synthesis of ATP energy. That's why it is powerhouse of cell. Here in mitochondria, this outer membrane, it is smooth, but this inner membrane which grows inwardly means this inner membrane shows infoldings, and these infoldings or finger-like projections are named crista. These are crista, and on these crista, the infoldings possesses tennis racket-like particles. These are elementary particles. These are F0, F1 particles. Or note down, these are ATPase. Means these are main center for the synthesis of ATP. These particles also named as oxysomes. These are oxysomes, F0, F1 particles. The number of mitochondria that depends upon the nature and activities of cell. More active cell having more mitochondria. As in case of chlorella, green alga, only one chloroplast. In case of spermatocytes, the sperm cell, only 25 mitochondria are there. In case of oocytes, about 30,000 mitochondria per cell. In case of kidney cell, which are less active, only 300-400 mitochondria, but liver cell more active, having 500 to 1000 mitochondria. The number increases as the cell becomes active. In case of giant amoeba, cavus, about 50,000 mitochondria, and the maximum number of mitochondria observed in more active cells like flight muscle cells of birds, about 5 lakh mitochondria per cell. Flight muscle cell in case of birds more active, more mitochondria. Per cell about 5 lakh mitochondria. Here in mitochondria, two chambers are there. One is outer chamber, and this outer chamber lies between this outer and inner membrane. This is outer membrane. This one is inner membrane. The space between both outer and inner membrane, that space constitutes outer chamber. And this matrix or core of mitochondria, that constitutes inner chamber. Here in this matrix or inner chamber, several molecules like carbohydrates, proteins, RNA molecule, as well as, not down here, important point is double-stranded circular DNA. And this naked circular DNA makes the mitochondria as semi-autonomous cell organelle. Dear students, when the number is required, the mitochondria, they undergo divisions due to presence of this DNA. So, these are named semi-autonomous cell organelles. These are present inside the cell, but having power of division due to presence of this naked circular double-stranded DNA, as well as RNA, ribosomes, but not down here in this matrix. The ribosomes are 70S type. 70S type. Later on, I will discuss ribosome. Then I will discuss two types of ribosome, 70S and 80S. Here in this matrix, the ribosomes are 70S types. Like prokaryotes, like bacteria. Means these mitochondria may be free-living aerobic bacteria. But these are present inside the cells, having their own DNA, having power of multiplication. So these are semi-autonomous cell organelles. This inner membrane, which shows infoldings, and these infoldings, finger-like projections in this matrix, are crista. These are crista. Foldings, infoldings, ingrowths, finger-like projections are crista. And the space between this crista, that is intracrystal space. And in this inner membrane, this is chain of respiratory enzymes present. It means glycolysis takes place in cytoplasm, because the enzymes which required for glycolysis are present outside mitochondria, present in cytoplasm. That is anaerobic process. But here in this matrix, all enzymes which are required for Krebs cycle, TCA cycle, are present in this matrix. Krebs cycle takes place in this matrix, in this core, or in this inner chamber. But another important question, in this inner membrane, chain of respiratory enzymes present, and due to presence of chain of respiratory enzymes, electron transport chain ETC takes place here in this inner membrane. Note down, glycolysis outside mitochondria means in cytosol, cytoplasm. Krebs cycle in this matrix, in this core, in this inner chamber, and electron transport chain in this inner membrane, because the enzymes which are required for ETC present in this inner membrane of mitochondria. Due to presence of this naked double-stranded circular DNA, the mitochondria becomes autonomous, semi-autonomous cell organelle. The mitochondria shows two states. One is active state and another is inactive state. In active state, that is a condensed state. All NEET students, note down each and every point. In active state, this matrix becomes condensed, reduced, while this outer chamber, that becomes enlarged, and it occurs at the time of Krebs cycle and oxidative phosphorylation. And in inactive state, that is orthodox state. In inactive state, this matrix becomes enlarged and this chamber gets reduced. Two states of mitochondria, one is active, that is condensed state. At that time this matrix becomes reduced, but this outer chamber that becomes enlarged, and that active state appears during Krebs cycle, oxidative phosphorylation. But when mitochondria becomes inactive, at that time this matrix becomes enlarged and this outer chamber gets reduced. Here in this inner membrane, these tannous-like particles, elementary particle, oxysomes or ATPase, means these are main center for the synthesis of ATP. And each oxysome, ATPase, elementary particle, having three parts. This basal part, that is F0, and this small stalk, that is about 5 nanometer long. This basal part, which is F0 subunit, it is 11 nanometer long, and its head, which is F1 subunit, and this head or F1 subunit, its radius is about 8.5 nanometer. Mitochondria perform several functions. Synthesis of ATP, that is a main center of synthesis of fatty acids, synthesis of amino acids, several intermediates for the synthesis of biomolecules like chlorophyll, like steroids, like alkaloids, as well as mitochondria store and release calcium ions when are required. This is all about mitochondria, which is double membrane bound cell organelle having two chambers, outer chamber, this entire matrix, it is inner chamber. In this inner chamber, Krebs cycle, oxidative phosphorylation that takes place in this matrix in Krebs cycle, aerobic process in presence of oxygen, breakdown of food, oxidation of food to release ATP. For the synthesis of ATP, these elementary particles, F0, F1 particles, their number in per mitochondria about 1 into 10 to the power 4 to 1 into 10 to the power 5 per mitochondria. And this mitochondria, main center for the synthesis of ATP, several intermediates for the synthesis of biomolecules like steroids, alkaloids, chlorophyll. Here synthesis of fatty acids, synthesis of amino acids, and these are main storehouse of calcium ions, as well as the mitochondria release calcium ions when are required. This is all about mitochondria. In tomorrow's lecture we will discuss chloroplast. Thank you.